Hello everyone, I'm Koyun. It's been a while since I upload my last video because I've been having a series of exams since March. Therefore, I'm currently in a sem break, so I have the time to finally teach you on what is actually in Form 4 Chapter 2, the basics of it. Okay, today we are actually going to learn what are the cell components inside. I'm going to introduce you and what are actually the functions of it and the difference between the animal cell and plant cell. I bet you have already learned this before in your Form 1 science, but we didn't learn much in detail. But there are still some basics to it. So the biology one, whereby we will add on more to it. I hope you won't get traumatized by this one. It's just a beginning of biology. I know there are a lot of cell components to learn, but don't worry. As soon as you learn the more of it, yeah, and uh, repetition will actually makes you adapt to it and get used to it easily lah. So before I would like to introduce their function, I'm going to introduce you how to actually identify which organelle is which organelle. These are all the organelles that work behind the scene in your animal cell. So first of all, the most the main one lah, the largest one is actually the nucleus. So normally when you want to locate a nucleus, you aim to find the one that is a darkly stained circular region. This so nucleus contains this nucleolus, which is this darkly stained region. This is actually called nucleolus. But the circle that's surrounding it, that one is a nucleus. So this huge thing right here is a nucleus. Inside this darkly stained region, yeah, it is a nucleolus. But normally in our secondary school Form 4 biology, sometimes the diagram they have given you, they just directly put this whole thing as a nucleus lah. But if you want to be more accurate, inside the nucleus there's a darkly stained one. That one is only the nucleolus. But normally they would just straight out show a darkly stained region, then that's probably the nucleus to you. So this, to be specific, this is a nucleus. Yeah, inside the nucleus, it has nucleolus, but we didn't learn much in detail about nucleolus. That's why the very simple diagram they show you, they normally just put in a darkly stained region and they just ask you what organelle is this, what cell component is this lah. And other than that, we are going to identify the whole process inside an animal cell. You need energy, right? So who's the one that supplies the energy? It is the mitochondrion. So this is the mitochondrion. A mitochondrion is in a singular form. Mitochondria is in a plural form. So this is only one. That's why I'm only writing it as mitochondrion. So it's the one that generates energy in form of ATP through cellular respiration, whereby you will learn in detail in chapter 7 for 4. Okay, other than that, these are the three things students always get confused all the time. Why? Okay, there is rough endoplasmic reticulum, smooth endoplasmic reticulum, and Golgi apparatus. So how do I actually identify what is rough endoplasmic reticulum? Normally, most of the time, rough endoplasmic reticulum is connected to the nucleus. Can you see that it is connected to your nucleus for this rough endoplasmic reticulum? Other than that, another thing you can identify is it has a ribosome attached to it. So can you see the black color one I drew? Yeah, those are ribosomes that attach to the surface of rough endoplasmic reticulum. So how do you identify? First of all, you see, is it connected to nucleus? So if it's the one that connects to the nucleus, it is most probably your rough endoplasmic reticulum, rough ER, I put it right here. And if you can see whether the surface is it smooth or does it have anything on it? Okay, so if it's not smooth, it is most probably your rough ER. What does it mean by not smooth? Rough? Why is it rough? Because it has, okay, does it have ribosome attached to it? So if it has a ribosome that attached to it, it is a rough endoplasmic reticulum. Okay, so this is a rough ER, but we must write it in their full name, reticulum. Be aware of their spelling also lah. And the one attached to the surface of it is called the ribosome. Okay. Other than that, rough endoplasmic reticulum has a sibling. Okay, it's called a smooth endoplasmic reticulum, the opposite of it lah, smooth. So it's very obvious the surface of it will be smooth, right? But a lot of students get confused between smooth endoplasmic reticulum and Golgi apparatus. There's actually an easy way to identify and differentiate between them. Hey, can you see the difference between Golgi apparatus and smooth endoplasmic reticulum? Normally, Golgi apparatus, it looks like a Wi-Fi. Why do I say it looks like a Wi-Fi? Okay, you know the Wi-Fi symbol? It looks like that. Ding ding. Yeah. So it kind of looks like a Wi-Fi. And the most important thing you can differentiate is it is not connected between them. Can you see the difference here? This layer is connected to another layer is connected to another new layer, and yeah, basically they are connected. But for Golgi apparatus, do you see any connection between them? The connection, right? Don't have any connection. And normally, if they want to hint you, they will actually draw out this vesicle. This is actually a vesicle lah. But it normally won't ask what is this lah. It's basically a vesicle that came out from the Golgi apparatus. It is a transport vesicle. Wait, is it a transport or secretary? Okay, so sorry. It's actually a secretary vesicle that because normally Golgi apparatus is the one that secrete out a secretary vesicle to be transported out. But that one we will learn further in detail in the production of extracellular enzyme lah. Because transport vesicle is actually from the rough endoplasmic reticulum. There are two types of vesicle if you want to learn more in detail lah. Okay. Other than that, this is how you see these two differences. What am I talking? Oh, yeah. Smooth endoplasmic. Okay, now we are done with identifying the rough ER, smooth ER, and Golgi apparatus. Okay, Golgi apparatus, yeah, they do produce a vesicle to transport proteins out of their cell, and they do also synthesize lysosome. So what is a lysosome? Lysosome is something, it also looks like a vesicle, but normally students would just identify them as lysosome lah. Okay, so yeah, lysosome is derived from a Golgi apparatus. It when the Golgi apparatus, yeah, it buds off a vesicle, and it can be a secretory vesicle or it can be a lysosome lah. So when they ask you to identify which is a lysosome, you just find anything that looks circular. Yeah, these are all lysosome. They won't ask what is the transport or secretory vesicle lah. So most of the time, this is just straight up a lysosome. You don't have to worry is it a vesicle or not. They are both look the same. Okay, basically it is in that way. Other than lysosome, we also learn something that is involved in the cell division, which is this one. It is called a spindle centriole, sorry, centriole. So this centriole will actually synthesize spindle fiber during cell division. These are all the cell components you have to know. And what about our plant cells? So we just basically revise again. For plant cell, what are the difference between them? How to differentiate? Basically, animal cell, you can see that they have irregular shape. It can be oval shapes, yeah, slightly circular shape. But for this plant cell, it's normally like rectangular shape. It has a regular shape. And other than that, there is two layers surrounding it. So the outermost layer is called the cell wall, whereby animal cell doesn't have it. And the inner layer of it is called the plasma membrane, where we also have it in our animal cell, plasma membrane. Okay, other than that, yeah, the one I drew in blue in color is something animal cell doesn't have. So what are these? These are actually the one that is very huge, okay, larger than the nucleus, a bean kidney shape like that. That one is a vacuole. And other than that, you can see this one, it has a thylakoid inside, granum inside. Yeah, it is chloroplast. Something that doesn't look like a mitochondrion, but it's also like a spherical shape. That one is a chloroplast. Mitochondria, normally they will draw it in this way lah. It's like in this pattern. Yeah, because yeah, oh my God. Yeah, basically it looks like that. So this one is chloroplast. We'll learn in detail in Form 5 lah. Inside, they have granum and thylakoid. That's why it looks in this way lah. Okay, let's revise back what we actually learned in just now. Just now. So the one that is have the darkly stained region, that one is the nucleus, right? And what is connected to the nucleus? Rough endoplasmic reticulum. So rough endoplasmic reticulum has a ribosome attached to it. So if you want to make sure, yeah, other than attaching to the nucleus, there is a ribosome that attached to it. So it is a ribosome. This how you identify a rough endoplasmic reticulum. Okay, and the one that is not connected. This one is not connected to each other, right? This is the Golgi apparatus. And the one that is connected to each other, yeah, this one is basically your smooth endoplasmic reticulum. You can see the outermost surface, the outer surface of it is smooth also. Another thing you can make sure. And last but not least is your lysosome. So lysosome is normally in a, yeah, circle shape kind of lah. They normally won't ask you to label a vesicle. We normally learn it as a lysosome. Lysosome is basically a vesicle that is part off from the Golgi apparatus lah. Yeah. Anyway, yeah, last but not least is your mitochondrion. So how do I actually know the function of it? How do I actually memorize them? So for the function of them, you actually have to relate. In order to memorize easier, you try to relate the process of synthesizing an extracellular enzyme, let's say lah, or synthesizing a protein. So let's say if you are a protein, what do you actually need? What are the ingredients or where are you born from? So if you're a protein, first of all, you need a DNA because DNA consists of the genetic code, genetic information that codes for the protein. And this DNA, it will transcribe into mRNA. and this mRNA will travel to the ribosome. Ribosome is where the site of protein synthesis. And this protein synthesized by ribosome will be transported by rough endoplasmic reticulum as a vesicle. And then once it reaches the Golgi apparatus, it will actually be modified. And yeah, it will Golgi apparatus have this ability to protect processes, modify, and pack, and last but not least, transport the protein synthesized through the secretory vesicle. So this is how a protein, yeah, synthesis of protein occur, like synthesis of protein occur. Ta-da! So here are the process of how a protein is synthesized. So in this way, you can try to relate what are their function instead of memorizing them individually and forcing them in your brain. So you learn it in this flow manner. Okay, inside the nucleus, it contains DNA. Okay, so yeah, this is one of the function of your nucleus, and it also control all cell activities. So this DNA consists of the information needed to be transcribed into mRNA. Then the mRNA will travel to the ribosome for the translation of protein. So that's the reason why nucleus contain this DNA. And then, yeah, this ribosome will synthesize the protein. And after the protein are synthesized, it needs someone to transport the protein, right? So rough ER is the one that transport the protein synthesized by ribosome. So this is the function of rough endoplasmic reticulum. But you don't write it as ER. You have to write the full name of it. And after this protein are transported to the Golgi apparatus, this Golgi apparatus have to be the one that modify the protein because a protein, they are very general. In order to let them become a functional protein like an enzyme, where we will learn it in chapter 5, you need to kind of modify it, change its shape, because the shape will actually determines how it functions. It can be a different type of enzyme, a more specific with different shape. That's why you need this Golgi apparatus to actually fold the polypeptide, which is a long chain of the amino acid lah, for this protein to become more specific, to have their own specific function. So Golgi apparatus have this function on processing, modifying, and pack and transport the protein. Because some of the protein, like extracellular enzyme, it has to be transported out of the animal cell. That's why it has a function to transport the protein. So these four is easier way for how, I mean, it's a flow for you to memorize their function lah, basically. And what about the others? So mitochondria is very obvious. It is the one that is a powerhouse of the cell. Yeah, it will be the one that generate energy in form of ATP through cellular respiration. Cellular respiration. We'll learn this in detail in the Form 4 Chapter 7 lah. Okay. Moving on, we also learn another one which is called the lysosome. What does a lysosome do? Yeah, lysosome is actually made from the Golgi apparatus lah. It actually synthesized lysosome, but we didn't learn that in detail. I mean, we didn't learn this info lah. But basically, what do lysosome contain? It actually consists of digestive enzyme. Since it has a digestive enzyme, it's very obvious. It's the one that is the in charge of breaking down, hydrolyzing damaged cell or breaking down the bacteria. So it's like a rubbish bin lah, I could say. Like it's the one layman term. Don't write it as a rubbish bin. So it's the one that hydrolyze damaged cell component. Let's say your mitochondria already rosette. Okay, your mitochondria very old already. Okay, so this lysosome, it will go and, yeah, basically engulf it because it has this digestive enzyme to dissolve, to hydrolyze them, break down them. Yeah, hydrolyze them more damaged cell component, or you can say break down the bacteria. Why? Because it has a lot of enzyme, digestive enzyme. And plasma membrane, yeah, it has a lot of function. Basically, plasma membrane is the one that separate content of the cell from external environment, separate cell content, yeah, and control movement of substance in and out of the cell lor. Okay, and we will learn this in detail during this chapter 3. Okay, and last but not least, when an animal cell wanted to divide, okay, divide into two, yeah, that's called cell division lah, via mitosis. So there's actually something that's very important that is involved in it. It is called a centriole. So what does this centriole do? It will actually form a spindle fiber during cell division. And for this one, you actually will learn in detail in the chapter 6 lah. Okay, so these are the basics of it for the animal cell, for their cell component. And I totally forget something, which is your smooth endoplasmic reticulum. So what does your smooth endoplasmic reticulum? Since rough endoplasmic reticulum is the one that transport protein synthesized by ribosome, smooth endoplasmic reticulum, meanwhile, it is the one that synthesize and transport lipid. Other than this function, it also detoxify drugs and metabolic byproduct. So what is a metabolic byproduct? It's basically when an animal cell or when a cell wants to carry out a metabolism, a process, it will actually produce some products that are not really required by the cell itself. So sometimes this metabolic byproduct can be toxic to the own cell. Therefore, this smooth endoplasmic reticulum will actually detoxify it. And the drugs also the same thing lah. It may harm your own cell. That's why the smooth endoplasmic reticulum, it will be the one that detoxify it. But the main function is the synthesize and transport lipid lah, basically. So that's it for animal cell. And for plant cell, you have to remember that they do not have centriole. It's something very special about them. Even though they don't have centriole, they can form spindle fiber. It's something super, yeah, interesting to know. Okay, what about this chloroplast, where plant cell have it, chloroplast and vacuole? So chloroplast is basically the one that is involved in photosynthesis. Yeah, chloroplast is basically, it has this pigment, green pigment. The pigment that makes the plant cell green in color is actually not because of chloroplast. It's because chloroplast inside consists of this thing called chlorophyll lah, in specific lah. Yeah. So it has a chlorophyll that actually absorbs sunlight and converts to chemical energy during photosynthesis. Yeah. Basically, chloroplast is the one that involved in carrying out photosynthesis. And what do the vacuole do? Vacuole normally is the one that store it. Yeah, it store the cell sap lah. It is a liquid filled sap. So this cell sap contains water, nutrients like, yeah, sugar, amino acids, got mineral salts, and so on. Other than that, it also have this function on making this plant cell turgid when it is filled with water. Yeah. And last but not least, our jelly-like medium. whereby you call it as cytoplasm, which also present in both animal cell and plant cell, is the medium whereby biochemical reaction will occur inside the cell. Yeah, so that's all. And for the cell wall, we also, yeah, cell wall is the one that maintain the regular shape of the cell lah. So maintain the shape of your plant cell. And it also provide mechanical support to the plant cell. Oh, I'm blocking it. Provide mechanical. And the very easy